Welcome. Tonight's show is From Signals to Survival. It's going to be an overview of cellular communication. And I am so happy to see the folks who are chatting in. And that is the great thing about YouTube Live. Go ahead, ask questions. It is fantastic to see people here. So let's start with letting us know what this show is all about. And here's what you need to know. Everyone's had the experience where you get startled, scared. You're walking through a parking lot and all of a sudden there's a dog that is inside a car and the window's partially down and it starts snarling at you and your heart starts beating. That is all about a hormone that's called epinephrine. Epinephrine, also known as adrenaline, and it's a huge part of the fight or flight response. The fight or flight response is something that occurs throughout your entire body. You'll have decreased digestion, increased heart rate, your pupils will dilate, your bronchioles, those tubes in your lung, they'll dilate. And what we're going to focus on largely tonight is what happens in your liver. What happens there is that epinephrine works to take a polysaccharide that you've learned about in Unit 1, glycogen, and convert it into glucose, a monosaccharide. So the question for tonight is, how did that happen? How did a substance that was released in one part of the body have an effect on another part of the body? That's what we're gonna talk about tonight on the AP Bio Show. And I am so happy that so many of you are here watching. This is really awesome. And on the show tonight, we'll be talking about cell communication as our lesson of the week. That's probably coming up in just a week or two. For those of you who are in an AP biology class, you're probably working on cellular respiration photosynthesis now. We'll have a song of the week, my song, Cellular Communication. There'll be a weekly quiz, and there'll be tips for learning the greatest subject of all, which is biology. I am Glenn Wokenfeld, also known as Mr. W. I'm a retired AP biology teacher, and my mission is to help you succeed in AP, IB, or college biology. I do that through this show, but I also do that through my website, learn-biology.com, and my app called Biomania. I'm going to tell you more about those later. Before we get deeply into the show, I want to let you know that I've put together a checklist. It's called the Ultimate AP Bio Study Checklist. And it'll basically tell you what to study about when you're learning about cell communication, when you're getting ready for that test on cell communication, or when you're studying for the AP Bio exam. So go ahead and download it. You download that by going to apbiosuccess.com/checklist. And that it's yours to help you study, to help you succeed. Let's start. Cell signaling. We're going to start with the big picture. I am going to change my view so it's a little bit more like this. Fantastic. And I'm looking at the chat and I'm seeing all these fantastic names. And what would be great to know would be where people are coming from. I see a couple of teachers' names here, and I'm interested in cities. So if you could chat in, what city are you watching from? Then that would be fantastic. I'd just love to know where people are coming from. And again, I'm so grateful to you, wherever you are, for taking your time tonight to watch the AP Bio Show. So let's see. I got Concord, North Carolina, Charlotte, North Carolina. That's fantastic. And anywhere else? Someone's from Antarctica. Fantastic. A lot of folks from North Carolina tonight. That's good. All right. Well, welcome and fantastic to have you all with me. Cell signaling, the big picture. So the basic idea is that cell communication is not some weird thing where cells are normally on their own, not talking to one another. Cells are not shy. Cells are constantly in communication with one another. And that communication takes two basic forms. There's direct cell to cell communication, and there's also communication via signals. Those signals are also called ligands or ligands. Those signals might be classified as hormones. That's when they travel a long distance from one part of the body to another part of the body. Or there are local regulators that basically just move short distances. There are also other kinds of signals that even leave the body. Those are called pheromones. They're fascinating. They're not something we're really going to talk about tonight. Direct communication in plants and animals is something that every AP Bio student should know about. In plant cells, there are these junctions between cells that are called plasmodesmata. These are bridges. They're actual connections involving the cell wall and the membrane. You can see how there's a break in the cell wall, and the membrane would be wrapping around here, and they allow signaling molecules, along with water and other substances, to diffuse from cell to cell. So that's how plant cells can send messages from one cell to another. In animal cells, there are structures that are called gap junctions, and those gap junctions are built from protein channels. And you learned about protein channels in Unit 2 of AP Biology. Those protein channels, those gap junctions, allow substances, including signaling molecules, ligands, to move from the cytoplasm of one cell to the next. And that enables this cell to induce a change in this cell, because that's what communication is all about. It's about transferring information, it's about creating changes. I've used this term ligands a couple of times, and now let's expand on that a little bit. Ligands are signaling molecules, and what they do is they bind with receptors in a target cell and have some kind of effect. Many ligands are hormones, and you've been hearing about hormones for your entire life. Things like estrogen, testosterone, adrenaline, growth hormone. Those are all examples of hormones that we talk about. And as I said before, what ligands do is they bind with receptors based on complementary shape. This idea of complementarity, it's not new. We'll talk about it more in a minute. But notice that the binding leads to some kind of cellular response, some kind of effect. That effect is going to be something that we'll talk about in a minute. My slides got a little messed up. Sorry about that. Here it was. The main thing that I wanted to emphasize is that the binding between ligands and receptors is specific. It's as specific as that connection between enzymes and substrates that you learned about in Unit 3. And you know that every enzyme has a unique substrate that it binds with because it fits with it. Well, it's the same thing with ligands and their receptors. So I represented this ligand over here as a kind of circle binding with this part of the receptor over here. Here, I'm using a little bit more geometry to show this. There's a circle that binds with this complementary shaped receptor. And here's a triangle which binds with this complementary shaped receptor. But you know that biology is really complicated, and that biology is based on molecules like proteins, which have extraordinarily complex shapes. So a more realistic depiction, still simplified, is something like this, where a ligand that looks like this is binding with the receptor. The first form of cellular communication that we're going to talk about is quorum sensing. And quorum sensing is represented by these two images over here. The basic idea of quorum sensing is that bacteria change their behavior based on their density. And where we see this is in the formation of biofilms. Biofilms are these films of polysaccharide that are dense with bacteria that build up in areas where bacteria are living. And again, if you think biofilm doesn't matter to me, it does, because biofilms are what form plaque on teeth, and plaque on teeth are what lead to dental cavities. And the reason why we do things like floss and brush our teeth is to get that biofilm off of our teeth. Now let's talk about quorum sensing, because the basic idea is that the biofilm won't form when the bacteria are in low density, but they'll form abundantly when the bacteria are in high density. How are the bacteria able to do that? Over here, we can see how. This is a representation of the bacterial cell. And what the bacteria do is they're constantly releasing signaling molecules that bind to cytoplasmic receptors. Here's one over here at number 3. Here's another cytoplasmic receptor at number 5. When the bacteria are in low density, there's not going to be very much binding of ligand to receptor. There just won't be that many ligands around. But when there are a lot of bacteria, then all of these receptors will start to be bound by the ligand. That's what we see on the right side of this diagram. What that will do is it will enable this receptor ligand complex to interact with a specific part of the bacterial cell's DNA. That's what we see right over here. And what that will do is it'll induce gene expression. So over here, we see something that looks like a polysaccharide at number 6 being produced. It's only produced when there's a lot of bacteria around. And the basic idea is that now we see a mechanism by which bacterial cells can communicate with one another through the release of a ligand binding with a receptor that causes genes to be turned on. And it's just a very, very basic example of cell communication, just to emphasize that all cells communicate. So there we go. I'm looking at the comments, and here we are. Okay, let's keep going. Cell signaling involves 3 key phases, and this is stuff that you absolutely have to know. The first phase is reception of a ligand by a receptor. The second phase is signal transduction. Now, signal transduction means that that signal is going to be passed from the membrane deep into the cytoplasm. It's as if somebody came to the door and asked for somebody who was inside your house. They weren't allowed to get inside your house. And so you go to the door and you say, "Oh, who are you here for?" And they say, "Oh, I'm here to talk to somebody." And you say, "Hey, someone's here to talk to you." That's transduction. You're taking the initial message and passing it deep inside the cell. And that leads to a cellular response. So that's the basic 3-part model of cell communication: reception, transduction, response. And we'll be coming back to this diagram throughout. So now let's break down each of those phases. We'll start with reception, then we'll look at transduction, and then we'll look at response. So in the first case, the signal molecule in reception, the signal molecule is going to bind with the receptor, and that receptor is embedded in the membrane. And as we said before, the binding is based on complementary shape. We talked about that earlier. That's reception. Now we're going to move on to the second phase, which is transduction, and the third phase, response. So what will happen is that the receptor interacts with membrane proteins to produce a second messenger. It's represented by this oval over here. That brings the message into the cytoplasm, and 2 things can happen. That cytoplasm is filled with enzymes, and there might be some kind of communication process that turns those enzymes from dormant to active, from off to on. Another possible response is that this signaling system, it's called the signaling cascade, will make it into the nucleus, and then it'll activate genes. Again, 3 parts to cell communication: reception, transduction, and response. M. Smith is asking a great question. How does the cell know to respond? The cell has a receptor, and the cell is essentially waiting for the ligand to bind with the receptor. It only responds when the ligand binds with the receptor, and that sets off transduction of the message. So, excellent question. Enzymes, Zans, I'm going to direct you to look on Learn Dash Biology, which I'm going to show you later. And I have a beautiful tutorial about that. And we'll come back to that a little bit later. And Grayson is asking me to go over the entire Krebs cycle and explain it in depth. Grayson, maybe I'll be able to get to that at the end of the show. But the main purpose of tonight was to talk about cell communication. And I do really want to appreciate the fact that people are now asking really good questions. And SVXID is asking, can the signaling cascade activate genes without going into the nucleus? Something has to go into the nucleus to activate genes. And what we'll see in Unit 6 is that there are molecules that go into the nucleus of a eukaryotic cell, and they activate a process called transcription. DNA makes RNA, makes proteins. So there is something that somehow has to go into the nucleus at some point. It doesn't have to be exactly like this, but there's going to have to be some pathway that does that. And in fact, the next slide that I'm going to talk about relates to that. I want to emphasize that everything that I've talked about so far relates to polar hormones. Remember, we've got things that are polar, that can interact with water, water soluble, hydrophilic, and then we've got things that are nonpolar. So all of this is about things that are polar, hydrophilic, soluble in water. And that's because those things can't pass through the plasma membrane, which is made of phospholipids. So they have to bind at a receptor. That's how polar hormones work. That is very different from steroid hormones. So polar hormones bind at the membrane, they activate second messengers. Steroid hormones work by a very different mechanism. And that mechanism is the following. Those steroid hormones act in a way that's pretty similar to what we saw with quorum sensing. And basically what those steroid hormones, what's an example of a steroid hormone? Ones that you just experienced as you went through puberty. Things like testosterone, the masculinizing hormone, or estrogen, the feminizing hormone. Those are non-polar. They can diffuse right through the membrane. And what they'll do is they'll bind with a cytoplasmic receptor. Those cytoplasmic receptors will then form a kind of complex, a receptor hormone complex. They'll diffuse through the nuclear membrane. They'll make it through a nuclear pore where they'll act as what I was just talking about, a transcription factor, and that'll lead genes to be transcribed into RNA. That RNA will make it out into the cytoplasm, and that will be then translated into protein. So steroid hormones make it into the cytoplasm, then they bind with the receptors, wind up activating genes, and that leads to long-term changes. But I do want to emphasize that there are some polar hormones that can also activate genes. They just have to do it in a slightly more complex way. Growth hormone is an example of that. Here's a gland. Here's a hormone. The hormone goes into the bloodstream. It circulates all throughout the body, every single part of your body. It only induces changes in cells that have the right receptor. In this case, it'll set up what's called a signal transduction pathway. That involves a second messenger that we just talked about and some signaling molecules. And then ultimately, the last member of that will be the transcription factor that goes into the nucleus. So let's see where we are. I want to emphasize that this is complex stuff. If you need some support, you can do tutorials about cell communication on learn-biology.com. And I am going to scan the questions. And here's a good question by Kara. Do cells ever misunderstand each other? Absolutely. Things can go wrong in all of these processes of cellular communication. I'll talk a little bit about that at the end of this show, but more importantly, when we talk about cancer, cancer is basically a breakdown in cell communication. It's when cells divide in the absence of a signal for cell division. So we'll talk about that in a couple of weeks. Already answered this question. Okay, fantastic. Okay, Want to emphasize that I think a lot of you are in classes that are already using learn-biology.com, and that makes me really happy because that's the best thing when teachers are using learn-biology.com. But if you're here on your own, I want to emphasize that you can subscribe to learn-biology.com, and it'll give you the skills and the confidence that you need to ace your class and the AP Bio exam. It's 100% aligned with the College Board. It's approved by the College Board as a textbook alternative, which is why hundreds of teachers are using it, with thousands of students around the world. It has interactive tutorials that have videos, flashcards, and quizzes that teach you everything that you need to know about every topic in AP Biology. The interactions include multiple choice questions, click and drag diagrams, identify the part diagrams, free response questions, and you get positive feedback for correct responses, and you get hints and repetition when you get it wrong. So if you work with Learn Dash Biology, you wind up understanding the material. There are student learning guides that tell you exactly what you need to do and how to work your way through the tutorials. And we have AP Bio review materials that give you everything that you need to crush it on the AP Bio exam. If you're a teacher, you have student performance monitoring, a kind of grade book, so you can see what work your students are doing and what work they aren't doing. And there are free trials that are available for teachers and students. And again, if you're here watching because your teacher directed you to, then you are already set. Those of you who aren't, I want to encourage you, go to Learn Dash Biology. You can start with a free trial. What we're going to do now is we're going to do an illustrative example of cell signaling, one that shows up a lot on the AP Bio exam, and one where we'll be able to go into depth and really test our understanding of cell signaling. So let's go. We're going to talk about epinephrine and G protein coupled receptors. Let me say that again. Epinephrine and G protein coupled receptor systems. The context for this is what we talked about at the top of the show. It's the fight or flight response. You get scared, you get startled, you get psyched up, and you have this effect that goes all throughout your body. That response is largely related to a hormone that's called epinephrine. It's a key hormone in the fight or flight response. So here's epinephrine. Epinephrine is a kind of modified amino acid. This is the skeletal structural formula over here. And you can see this hydroxyl group over here, this hydroxyl group, this hydroxyl group, this amino group. It is polar. It's not going to make it through the cell membrane. So epinephrine, also known as adrenaline, it's polar, water soluble. It's released from the adrenal glands. The adrenal glands sit above your kidneys. Epinephrine's effects are widespread but tissue specific. So, as I said earlier, epinephrine goes into the bloodstream from the adrenal glands. It goes everywhere. Some cells don't have receptors for adrenaline, for epinephrine. They don't do anything. Some cells do have receptors, and then what those cells do based on the kind of tissue is based on the kind of tissue that they are. So in the stomach, epinephrine will cause digestion to decrease. In the heart, it'll cause the heart rate to increase. It'll cause your pupils to dilate. It'll cause your bronchioles to expand. We're going to focus on is this over here. In your liver, it's going to cause glycogen to be broken down into glucose. And remember from unit 1, glycogen's a polysaccharide. It's made of glucose monomers. So what you've got to do is you've got to break all these bonds between the glucose monomers so they can be released into the bloodstream. Why do they have to be released into the bloodstream? That's all about unit 3 of AP Biology, cellular respiration, so that glucose can power cellular respiration and make ATP. And this is what I just said. Epinephrine induces liver cells to hydrolyze glycogen, a polysaccharide, into glucose. The glucose diffuses into the blood, provides energy to fight or flee. And our question is, how does epinephrine induce the glycogen to glucose conversion? That's what we're going to focus on tonight. I'm going to scan for a couple of questions. So I'm seeing a question that's really sort of a strategic question about how to go through the curriculum. Um, and SVXID is saying that my teacher makes us finish a unit of AP biology in a week. I don't think you're talking about a whole unit. I think maybe you're talking about a topic. Um, and here's the problem that AP biology teachers face, and it's a rough problem. AP biology is huge. This won't make you feel any better, but it used to be a lot huger than it is now. And you just have to stick to a pretty rigorous schedule in order to make it through the course. So I know that that's small solace for those of you who are probably spending hours every night. The only thing that I can say is that biology is the coolest subject. It's worth it. You're going to learn so much, and you just got to stick with it. So I don't. Uh, I don't know if I have anything better to say about that. I do see a question from Zaid. How does the body know when to release epinephrine? I'm going to go back a slide or two. Okay, so here's your brain. Your brain has an experience where it gets scared, startled, someone sneaks up on you and says boo, a dog barks at you and you didn't expect it. So your brain perceives the threat. You can hear it, you can see it, maybe you can even smell it, you know. And in response to your brain perceiving the threat, there'll be a message that gets sent down through the nervous system, which is really outside the bounds of AP biology. But you have a brain, you have nerves, you have a spinal cord, and there's going to be a nerve that's going to go down to the adrenal glands. And those adrenal glands, in response to that nerve impulse, will release epinephrine, adrenaline. So that is the response to Zaid's question. And Anna is asking, without epinephrine, glycogen can't be broken down at all? No, that's not correct. What I'm saying is that this is how glycogen gets broken down as part of the fight or flight response. But here's another thing that happens. Every time you get hungry, your body releases another hormone called glucagon. And that glucagon goes into the liver and works in almost exactly the same way as the pathway that we'll be talking about tonight. So when we learn about feedback and homeostasis in about a week, you can easily reapply what you've learned tonight to that subject. And SVXID is asking, is the liver the only organ that can hydrolyze glycogen? When you eat a carbohydrate rich meal, you'll store your glycogen in a couple of places, but the most important of those are the liver, but also muscle tissue. And between the two, you can store a couple of pounds of glycogen, like some small percentage of your body weight. And when you're needing energy, that's how your body will, that's one of the first energy sources that your body will turn to. Again, we're getting into some really cool stuff related to sports physiology. Let's go on. Talked about this. Okay, we talked about this. Beautiful. So here's where we are. And here's where we're going to get into some beautiful biology details that I hope excite you as much as they excite me. I've been teaching myself this stuff and learning it for my entire life, and I hope you find it as stimulating and kind of as inspiring as I do. This is a beautifully intricate process, kind of like glycolysis and the Krebs cycle and the electron transport chain, photosynthesis. These are all beautiful processes, and we're about to learn about another one. So the way that epinephrine induces this glycogen to glucose conversion is through a G protein coupled receptor system. So here's what it looks like: a G protein coupled receptor system that induces a phosphorylation cascade. Your being able to actually say the sentence that I just said could get you a couple of points on an FRQ. So I'm going to say it again. The way that epinephrine works is through a G protein coupled receptor system that induces a phosphorylation cascade. Good for you to know that. Now let's take apart what that means. G protein coupled receptor systems in liver cells look like this. So my text got a little cut off. Sorry about that. But here are liver cells. Here's the liver. Here are some cells represented by this hexagonal shape. And here's the membrane of those liver cells. Here's the membrane. Okay, so now you can see the phospholipid bilayer. So here's the resting state when there's not epinephrine. The receptor's not bound by the ligand. There's no epinephrine in the system right now. There's a G protein nearby. Why is it called a G protein? It's called a G protein because of the way it interacts with GTP and GDP, I believe. I'm not 100% positive of that. But note that we have the system where we have a receptor, a G protein, and a nearby membrane-embedded enzyme that's called adenylyl cyclase. And that is not an easy thing to say. I thought it was a typo the first time I saw that, but that's what it's called, adenylyl cyclase. So no hormone. This is in its inactive off state. This is in its inactive off state. And therefore, the second messenger that's going to turn this system on is not activated. Okay, that's when the system is off. Now you have this scary thing happen to you and you release epinephrine. So epinephrine, which is being represented by this circle, I chose not to use a structural formula just to keep things simple. What happens is that the epinephrine binds at this binding site on the receptor, and it induces a change on the other side of the receptor. And that's what I just said. Epinephrine binds with a G protein coupled receptor. The receptor changes shape on its cytoplasmic side, and the nearby G protein is still dormant. When it's dormant, it's bound to GDP. GDP stands for guanine diphosphate. And you've already learned about ADP and ATP, where ADP is the low energy form and ATP is the high energy form. It's exactly parallel. Let's look at this receptor in a little bit closer way. Here's the ligand binding site, binds with epinephrine. There are 7 alpha helices. You learned about alpha helices back in Unit 1 when you learned about protein structure. And then there's this part over here that extends into the cytoplasm. And the thing to know is that when something binds over here, the binding can induce a ripple effect that ripples through these alpha helices and causes a change over here. So after epinephrine binds, it induces a change over here, and that activates the G protein. The G protein, it's not like it's moving purposefully, it's sort of randomly moving in the membrane. It'll bump into this part of the receptor, and that will induce a change in the G protein, and that'll cause the G protein to let go of this that'll cause the G protein to let go of GDP and bind GTP. It's now in a high energy state. Why? Because of epinephrine binding with the receptor. The G protein is now activated. Now we're going to watch how the next step occurs. That activated G protein bound to GTP drifts in the membrane until it bumps into adenylyl cyclase. And again, these things aren't purposeful. There's random motion, but there's a complementary fit between the G protein and adenylyl cyclase. And when the two bind, adenylyl cyclase, which was an inactive enzyme, becomes an active enzyme. And it will grab an ATP in the cytoplasm and convert it into a related molecule, cyclic AMP. Cyclic AMP is the second messenger. Now, there's a lot of stuff that always comes at you in biology, and you just kind of have to take a deep breath and rely on what you know. You know that ATP can be converted into ADP. You've seen that happen during cellular respiration. During cellular respiration, you've seen that happen in various processes where ATP is providing the energy for example, a dehydration synthesis reaction. Well, in this case, what we're doing is an enzyme is taking ATP and converting it into cyclic AMP by chopping off these two phosphates over here. Here's a closer look. Here's ATP. Here's adenylyl cyclase. I didn't show it in the membrane. I'm just showing it in the typical iconic enzyme form. And it takes cyclic ATP, chops off two phosphates, and creates cyclic AMP over here. Here's the structural formula. It's called cyclic AMP because of the way that the phosphate is bound to the adenine over here. What do you need to know? Cyclic AMP is a modified form of ATP. It's the second messenger. All right. I'm just going to check the comments over here. Okay. Delbers is asking a great question about how did someone even discover this stuff? And in this case, unfortunately, I don't know, but I'm going to make a little note about that because that is a really, really cool question that I want to follow up on, which is the discovery of ATP. I'm just making a little note here. I said the wrong thing. Discovery of cyclic AMP. Okay. And Jace is saying, so GDP is parallel to ATP in terms of power or function. You said that in the wrong way, but I think you're going in the right direction. GTP is parallel to ATP in terms of power or function. And that's the thing to remember. So, like, for example, when we were talking about cyclic AMP over here, or actually more importantly, when we were talking about the activation of the G protein, I'm going to go back a little bit more. Low energy, high energy. When this is bound to GTP, it's energized. When it was bound to GDP, it was essentially off. Just doing a little time check here. Okay. So here we were, did all this. Fantastic. Okay. So I've got a slide for review, and this is really a great time to ask questions. And I see one question by Pradishka. How does the body know how much epinephrine to release? I don't know that exactly, except for that I think that you release epinephrine. So there's like a pulse of epinephrine that gets released from your adrenal glands, and it'll continue to be released for as long as you're in that threatening situation. So, for example, a soldier in a war might be producing adrenaline for a really, really long time. But when the specific battle ends, then through a mechanism that is well understood, adrenaline release, epinephrine release will come to an end. So, Pradishka, that was a cool question. And are there cases where you can't produce adrenaline epinephrine? That's a question for a physician, which I am not. I don't really know of any conditions like that. And Music and Night Vibes is asking a cool question. Is GDP used for any other processes? Yes. If you're in AP biology and now you're in unit 4, if you look back at the Krebs cycle, there's a moment in the Krebs cycle where the Krebs cycle is producing a little bit of ATP, but it does that in a kind of convoluted way where before it produces ATP, it produces GTP, and then another enzyme takes the phosphate from GTP and phosphorylates in ADP, making ATP. So cool question, Music at Night. And I actually think that all of these nucleotides, so there's like GTP, and I think there's actually, and when DNA and RNA are made, they're usually, they come in their triply phosphorylated forms. So they're sort of like the molecules come in energized. Very cool question. And Kay was asking, what year did people discover cells? Cells have been known about for a long time. There was a guy named Robert Hooke who was one of the first microscopists and I think discovered cells in like the 1600s. It was about the same time that Isaac Newton was around. I think that's 1600 something. Okay, so let's go on. Just want to emphasize that what we've learned so far is this. Everything I've talked about has just been this top part of the diagram, right? Up to the second messenger. And now what we're going to talk about is how relay molecules lead to the cellular response. And again, remember where we're going. The cellular response is about the release of glucose from liver cells in response to the release of epinephrine. Awesome questions. Thank you very, very much. And I was just asking for questions and you just asked them. So we're going to just move on past this. All right. So I've got a little diagram of this on the bottom left. And remember, we've got ligands that bind with receptors. And then we have a second messenger. And then we've got relay molecules that lead to a cellular response. What we're going to do now is talk about transduction and amplification. We've kind of done transduction insofar as we've released the second messenger cyclic AMP. But now we're going to go through all of this fantastic stuff in here. Here's what you need to know. Cyclic AMP, which is the second messenger, is going to activate a chain of relay molecules. Those relay molecules are called, depending on how you want to pronounce it, kinases or kinases. That activation involves something called a phosphorylation cascade. Do not freak out at the mention of a word like phosphorylation cascade. You know that ATP, has phosphate groups. A phosphorylation cascade involves adding phosphates, adding phosphates, adding phosphates. That's what we're talking about. So how does that work? Kinases are these relay molecules, and they're proteins that are activated when they're phosphorylated. So on the left side, we have unphosphorylated kinases. And what we see over here is that because of the presence of cyclic AMP, this kinase over here becomes an activated kinase over here. And once it's activated, it does something really cool. It'll activate the next kinase in the chain. Now, there's a couple of other things that I couldn't really convey in this diagram, but there's a level of specificity. In other words, this protein kinase 1 will only activate protein kinase 2. It'll grab a phosphate and it'll glom it onto protein kinase 2, which will activate protein kinase 2. And then protein kinase 2 will activate protein kinase 3. Protein kinase 3 will become activated. And this will go on in a long chain. And then at the end, we'll get to the terminal enzyme. The terminal enzyme, I'm just representing by the letters GP, but that stands for glycogen phosphorylase. And what that does is that's the enzyme that takes glycogen and snips its glucose subunits off to release glucose into the bloodstream. Notice over here that we had this phosphorylation cascade. But what I want you to start imagining now is that there are multiple phosphorylation cascades that happen in parallel with one another. So each step involves multiple activations, and the result is signal amplification, where one epinephrine binds, but that'll lead to the production of several cyclic AMPs by adenylyl cyclase. Each cyclic AMP will kick off protein kinase 1, and then you have to imagine a whole chain, and that'll activate so many glycogen phosphorylases. So literally, we've amplified the system. And the way to think about this is that your liver cells are ready to do this. They're ready to take their glycogen and instantly release it into the bloodstream so that it can support the body in the fight or flight response. That's an important adaptation that helps your cells survive. But they don't do it until the signal comes in. Once the signal comes in, those cells spring into action. Those enzymes spring into action. They release their glucose after breaking down glucose into glycogen into glucose. And ZE Watching Account is asking a really cool question. I do think that it is not good for you to be overstimulated by hormones like epinephrine, because what they're doing is they're causing you to constantly, for example, in this case, release glucose into the blood. You don't want to release that glucose into the blood constantly. You only want to release it when you have to. So I do think that there are medic imbalances that um, are not good for you, and that involve overproduction of these hormones. But again, not a doctor, not a physician. I do know that like chronic stress is really bad for you. And so chronic stress that leads your body to think that it's in fight or flight, but it's not, that is not going to be good for you. And Delbers is asking, so the brain controls all of this? Insofar as the brain is the one that perceives the threat, yeah, it's the one that starts the whole cascade out. So let's now take a molecular view at the response. We've got the cyclic AMP, and it is activating all these protein kinases by phosphorylating them. We finally get to the terminal enzyme, glycogen phosphorylase. And what that does is it takes glycogen and snips all these glucoses off so that the glucoses get released into the bloodstream. And there we have our response. So think about the cool ground that we've covered. We started with reception. We've gone through transduction, release of cyclic AMP, and then finally we saw a phosphorylation cascade that led to a response. So I'm just going to scan for questions. Blaze is asking, hypothetically, is there a case that this model is altered in any way? Would that strongly affect our functions? Yes, it would. And again, I've talked about how at the end of the show, I'll talk about a couple of examples where there's homeostatic breakdown caused by breakdown in these systems. It's a very general answer to a very specific question, but best I can do for right now. Okay, so we've activated this response, and let's say that we're a soldier on the battlefield, or we had some scary something happen to us, and then our brain tells us, okay, the threat's over. How do we turn this thing off? So the end of the threat leads to the end of epinephrine secretion. Why? Because the brain will send a signal to the adrenals, or maybe differently, the brain will stop sending a signal to the adrenals and say, no more need for epinephrine secretion. When that happens, the epinephrine is going to diffuse away from the receptor. That epinephrine was never tightly bound to the receptor. It fit there because it had a complementary shape. Maybe there was some hydrogen bonding. But your body's at 37 degrees Celsius, and something that's only connected by a hydrogen bond is not going to stick there forever. So adrenaline, epinephrine, is no longer secreted, and the epinephrine is going to diffuse away from the receptor. That is going to cause a change in the receptor shape over here. And at a certain point, that G protein will convert its GDP back to G. And at a certain point, the G protein will take its GTP, break off a phosphate, which will render it into GDP. So now the G protein is turned off. Now that the G protein is turned off, down to GDP, adenyl cyclase is going to deactivate. That is going to stop the conversion of ATP to cyclic AMP. So we've got no more second messenger. So that's the beginning of the shutdown of this system, right? The threat is gone. We're not going to produce any more cyclic AMP. But then there's also things that happen in the cytoplasm. And the thing to note is that there are also enzymes that are called protein phosphatases. And what they do is they take these phosphorylated kinases and they dephosphorylate them. They break their phosphate off and render them back into their inactive state. So again, you have to remember that like your cells are like a football team that is ready to go, waiting for the quarterback to say, hut, go. And when they do say go, then you have these phosphorylation cascades, but then the play ends and everybody goes back to the inactive waiting state. So the cell is capable of instantly turning on and turning off. And then the cytoplasmic signal transmission ends, and we're not going to make any more glucose from glycogen. Holy cow. We've now explained the entire process. And people have been asking me questions like this throughout, like what happens if something goes wrong? Something goes wrong, it's really bad. So for example, cancer is a breakdown of a signaling system that basically leads to cell division signals when there shouldn't be. What we're going to see in just a week or two is that type 1 and type 2 diabetes are also caused by breakdowns in signaling system. There's a disease called hyperthyroidism, also caused by a breakdown in the signaling system. And the bacterial disease cholera also involves exploitation of a G protein system that leads to massive dehydration, which can kill you, which at the same time simultaneously spreads the cholera bacteria that causes the infection. So yeah, bad things can happen. Scrolling down to see new comments and okay. And is it possible? Someone's asking if it's possible to die from type 1 diabetes. Absolutely. And I'll teach about diabetes in one of the upcoming AP Bio shows. So, folks, I have a song that I've created about cellular communication, and it summarizes a lot of what we've learned. And I'm going to play it for you right now. I'm going to go like this. And I'm going to go like this. Have you ever had a sudden fright? Perceived a threat, you're all geared up for fight or flight. Heart pounding, face flushed, hair standing up on end. It's partly from a hormone called epinephrine. Epinephrine's got another name, adrenaline. Secreted from adrenal glands, it's stuff in EpiPens. A shot can save your life. If you have an allergy involving sensitivity to nuts or stink from bees, epinephrine's polar, so it can't diffuse through the lipid bilayer. So instead, it induces a second messenger, it's cyclic AMP, which stimulates massive cellular activity. Let's learn how liver cells respond to an epinephrine dose. What they do is break down glycogen to glucose, which streams into your blood to give you energy required to respond to threats as you fight or flee. How does epinephrine get your liver cell to do this function? It's 3 steps: first reception, then transduction, then cellular response. In enzyme activation, we're learning about the process of cell communication. Cellular communication works through phases 3. Reception of a ligand, they're complementary. Second messenger released, transduction is a beast. Now amplified response until the stimulus has ceased. Today's focus is G protein coupled receptors. We'll leave the other systems for another lecture. Epinephrine's the ligand that with the receptor binds. It's a hormone, the hydrophilic kind. Ligands bind to receptors only if the shape matches its specific, based on complementary attachment. Epinephrine goes everywhere but only affects cells with receptors with which it can connect. Once epinephrine binds at the receptor binding site, the receptor changes shape on its cytoplasmic side. But just inside the membrane you'll find the G protein, which until epinephrine binds is dormant, asleep. In its dormant form, the G protein's bound to GDP, but epinephrine binding makes it bind with GTP. Then the G protein drifts in the membrane space till it bumps into adenylyl cyclase, a membrane-bound enzyme whose signaling functions activating a second messenger, setting up transduction. Transduction converts a message into another kind. Message 1 was a ligand, which came along to bind with the receptor, which in response woke up the G protein, which gets adenylyl cyclase to take an ATP and remove 2 phosphates, making cyclic AMP. That's the second messenger. It takes a message deep into the cytoplasm, where it unleashes a chain of enzyme action, a phosphorylation cascade. The message was received, it was then transduced. Next we'll see how cellular response gets produced. Cellular communication works through phases 3. Reception of a ligand, they're complementary. Second messenger released, transduction is a beast. Now amplified response until the stimulus has ceased. A phosphorylation cascade involves a chain of enzymes known as kinases. kinases. These kinases get phosphorylated, which means gaining a phosphate, which activates them. And what do kinases do when they're charged up with a phosphate? They find the chain 6 kinase and phosphorylated. This chain of activation is like falling dominoes, because that's how a phosphorylation cascade goes. And each kinase carries out multiple activations, causing massive signal amplification. The signal grows in strength with every kinase action, building in intensity a chain reaction. One epinephrine binds, causing many cyclic AMPs, each of which activates many kinase proteins. By the chain's last kinase, tens of thousands activated. A cellular tsunami as the chain is terminated. Glycogen phosphorylase is the enzyme at the end that makes glucose by hydrolyzing glycogen. So into the blood from the liver, glucose gets secreted, and the threat that started the story can be fled from or defeated. It's adaptive how your cells are poised to spring into action. Once they hear epinephrine's alarm, there's a reaction in all eukaryotes, from amoebas to acacias. G-protein couple receptors used in cell communication. Cellular communication works through phases 3. Reception of a ligand, they're complementary. Second messenger released. Transduction is a beast. No amplified response until the stimulus has ceased. Back to the ligand of the receptor. It only stays for a moment before it diffuses away. So when the thread is gone, the cascade gets shut down. With no adrenal secretion, the receptor's unbound. G-protein drops the phosphate and bound to GDP. It goes back to sleep, stops its activity. Bound to GDP, it no longer stimulates adenylyl cyclase, which no longer creates cyclic AMP, so the second message stops. Kinase phosphorylation quickly drops as other enzymes, protein phosphatases, clip off phosphates, turn off kinases. Glycogen phosphorylase stops hydrolyzing glycogen, so blood glucose normalizes. Liver cells return to their resting state. I love how G-proteins let cells communicate. If you like this song, please subscribe. Your comments and likes help our channel thrive. And check out our website, Learn-Biology. I wrote it for you. And there you. Okay, folks. Let me come on back over here. How about like that? Okay. Thank you so much for sticking with me through that song. I hope you liked it. And what we're going to do right now is we're going to do our weekly quiz. I know that we took a little bit longer than we thought, but I was frankly really delighted to see all the people who were here and all the questions. Let me bring up the weekly quiz, which is going to be super fun. And let me go here and here. Okay, so what I've done is I've gone to the Unit 4 review page in learn-biology.com. I'm going to set the screen like that, and I'm going to adjust the size of this window a little bit. Much better. Okay, so every unit in AP Biology on learn-biology.com has a unit review page. We're just going to do a couple of multiple choice questions. Let's go here. I'm going to log in. Good. Go like that. I think I'm in. Fantastic. So, cell communication. I'm going to choose the questions that we want. 7. We won't do all of them. Let's begin practice. Oh, tough question. Okay. I'm hoping people can see that, and I am going to move myself over here. Yeah, good. Okay. So, plasmodesmata, shown below, are channels through which plants can send chemical signals between one cell and the next. That's the diagram that we just looked at in our slideshow. Researchers are testing the effects of a drug that inhibits the formation of plasmodesmata on plant growth. They tested 2 concentrations of this drug on the average growth of the plant in 5 trials. Here's a table that's showing what happens after 5 days, 10 days, and 20 days. And which of the following is the best explanation for the effect of the drug? So here's how these questions work. We've got 4 choices, and I'm going to explain what's going on in the table. And there we go. So at 5 days, in the control group, which didn't get any of the drug, there was 2 centimeters plus or minus 0.5 of growth. In the group that had 10 microliters of the drug, There was also 2 centimeters, plus and minus 7. And then with 20 microliters of the drug, it was 1 centimeter, plus or minus 0.5. That's after 5 days. After 10 days, the control had grown 5 centimeters, the 10 microliter group 3, and the 20 microliter group 1. At 20 days, the control had grown 10. The 10 microliter had grown 5 centimeters with a margin of error, and the 20 microliter had grown just 1 centimeter. So, which of the following is the best explanation for the effect of the drug? You're going to put in an answer 1, 2, 3, or 4. 1. The drug makes it more difficult for molecules that stimulate cell division to move from cell to cell, inhibiting plant growth. 2. The drug makes it easier for molecules that stimulate cell division to move from cell to cell, stimulating plant growth. 3. The cell makes it easier for molecules that inhibit the cell cycle to move from cell to cell, inhibiting plant growth. And 4, the cell makes it more difficult for molecules that inhibit the cell cycle to move from cell to cell, inhibiting plant growth. So, folks, what do we got? Do we have a 1, 2, 3, or 4? Please put your answers into the comment thread, and I will click on the one that gets the most responses. And and I've got one choice for 4, one choice for A, one quote from my glycolysis song. Thanks, Bella. And I'm going to go 1 or 4 from music. So let's go with number 1. The drug makes it more difficult for molecules. Way to go. It's choice number 1. Without plasmodesmata, signaling molecules would be prevented from moving from one cell to the next. If these molecules stimulated cell division, then plant growth would be inhibited. So if you got it wrong, you study the answer. Again, if you're in a class, just go ahead and do the quiz, and you can spend as much time with this quiz as you want. Second question. Which of the following mechanisms of cell communication are not found in animal cells? Again, I want a 1, 2, 3, or 4 in the chat. 1, long distance signaling through hormones that bind to receptors in target cells. That was choice 1. Choice 2, direct cell to cell contact. Choice 3, transfer of signaling molecules via plasmodesmata. Choice 4, short distance signaling through the release of ligands that bind to nearby receptors. Which of these is not, I repeat, not found in animal cells or células de animales? I think that's right. Anyhow, I see a bunch of 3s. Holy cow, we got a bunch of superheroes in the crowd. Indeed, plasmodesmata are found in plant cells, not animal cells. Okay. During many cell signaling processes, the enzyme adenylyl cyclase converts the molecule below into an important second messenger. As part of this conversion, which bond is broken? Is it the bond at A, B, C, D, or E? Please put in a choice. Is it A, B, C, D, or E? Those are your answers. Let's see it, guys. I want to see an A, B, C, D, or E for this answer. And I got one A, got another A, got another A. Okay, let's choose A. No. Let me tell you why. Maybe I did a bad job teaching this. Remember that the signaling molecule is cyclic AMP. That A, I don't think I said this. That A stands for monophosphate, 1 phosphate. So if you break this bond, you'd still have 2 phosphates. If you break this bond, you'd be down to 1 phosphate. And then if you hooked this part to the sugar over here, then you'd have AMP. So the answer was B, not A. But this is the beauty of LearnDash biology. If you get it wrong, you get another try. Oh, good question. Epinephrine, also known as adrenaline, is produced by the adrenal glands. The structural formula for epinephrine is below. Diagrams S and T below are general models for signal reception and transduction. Here's model S. Here's model T. I hope that's big enough. I think it is. Which of the following correctly identifies and explains the model that corresponds to how epinephrine works? Okay, so we have choices and explanations. Choice 1, model S, because it shows epinephrine diffusing through the cell membrane and activating genes. That was the first choice. Choice 2, model S, because it shows epinephrine binding with a cytoplasmic receptor. Choice 3, model T, because it shows epinephrine. Let me say that again. Model T, because it shows epinephrine phosphorylating a relay molecule. And choice 4, Model T, because it shows epinephrine binding with a membrane receptor. What do we got? We have 1, 2, 3, or 4. You can go 1, 2, 3, or 4, A, B, C, D. What do we got here? I think we have 1D. I'm going to wait for some other people to give me their answers. Is it 1, 2, 3, or 4? Okay. Seeing a couple of choices. And I'm going to actually guide you through this. Think about what we just learned. Epinephrine is polar, water soluble. Water soluble things can't diffuse through the phospholipid bilayer. So that eliminates model S. So choice 1 and choice 2 are out. This third choice, model T, because it shows epinephrine phosphorylating a relay molecule, the epinephrine binds over here. It doesn't phosphorylate the relay molecule. So really what we're down to is model T, because it shows epinephrine binding with a membrane receptor. If we clicked on the right choice, you would have gotten a hint and we would have come back to it. Folks, you are awesome, but it is way over time. So we're going to go back to our slideshow. And your plan for this week? Your plan for this week, most of you are with a teacher, and that's a beautiful thing. I'm going to change my view over here. Your plan for this week is, to basically do what your teacher tells you to do. But if you're learning biology independently, then it's time to go through Unit 4. And just go to learn-biology.com, then courses, AP Bio, Unit 4. And then you would do these tutorials that would take you through much of the territory that I covered tonight, but it would take you through it better because you'd interact with it and you would learn the material super well. That is what it's all about. You got to interact with biology to learn it. That's what learn-biology.com is all about. We are done, folks. And I can't tell you how much fun this was, how much I appreciated being here with you. I do these shows live when I can, and I will let you and your teachers know. You can check the Science Music Videos channel, and you can see when I'm doing a live show. I think I'm going to be able to do one next week. This has been so much fun to have so many of you here. It was really awesome, and I hope you're loving biology because it's the greatest subject. I'll have another video coming up soon for those of you who are watching. And everybody, just be well. Have a fantastic week. I hope to see you here next week. And everybody just stay safe, and I'll see you soon. Everybody, be well. Thank you so much.